Hello, my dear students. So, today we are here to discuss briefly the topic DNA replication. DNA replication is a topic from your chapter Molecular Genetics or Molecular Basis of Inheritance, and this DNA replication is the key mechanism by virtue of which our cells double their DNA so that they can perform the cell division. So, the topic is DNA replication. DNA replication. DNA replication. DNA replication. So the topic is DNA replication. So what do we mean by DNA replication? As the name itself speaks, replication means duplication or doubling. So it is duplication of DNA. It is duplication of DNA, or we can also say that it is synthesis of identical DNA, identical copy of DNA. It is synthesis of identical copy of DNA from the parent DNA. From parent DNA, that parent DNA is also called as template DNA. That's also called as template DNA. Template DNA. So definition means it is duplication of DNA. It is synthesis of the identical copy of DNA from the parent DNA, what we also call as template DNA. So if we have this parent DNA, or what we call as template DNA, so what we have to do, we have to synthesize the identical copy from it, and this identical copy will be new DNA, which will be passed on to the new cell, Now, this DNA replication, why does it occur? This DNA replication occurs in nucleus. What is the site of replication? The site is nucleus. So it should be clear, where is this DNA replication occurring? It is occurring in nucleus. Now there are various properties of this DNA replication. Before we discuss the mechanism of this DNA replication, we should understand various properties of this DNA replication that provide characteristic to this replication process. So I am going to discuss briefly the properties of DNA replication, or the features of DNA replication. Properties of DNA replication. First property of DNA replication is that this DNA replication is a semi-conservative process. So first property is that it is a semi-conservative process. It is a semi-conservative process. It is a semi-conservative process. What do we mean by semi-conservative process? Or what do we mean by it is semi-conservative nature? So to understand this semi-conservative nature, you will take this DNA double strand like this. If this is a DNA double strand, and this yellow color represents the parental DNA. So during DNA replication, what will happen? These two strands will separate. So one strand is right here, another strand is like here. So these are the two separated strands. So during replication, these two strands will separate, and the new DNA will be synthesized on this half strand like this. New DNA will be synthesized on this single strand, on these single strands. So if you look at the new DNA, what do you observe? In this new DNA, this one is the parental strand. This one is parental strand. And this one is the new strand. This one is parental strand, and this one is new strand. And likewise, same is true for this strand. There is one parental strand and there is one new strand. So there is one parental strand and there is one new strand. So because in this new DNA we are observing that there is one parental or original strand and only there is one new strand. So 50% of the new DNA is from the parental DNA and 50% is the new molecule. That is why we call it semi-conservative, which means that half of the parental DNA in the new DNA is from the parents. So is from the parental DNA. So what do we mean by semi-conservative process? It means that one strand of daughter DNA, one strand of daughter DNA is retained from parent DNA, is retained from parent DNA. तो सिंपल मीनिंग है इसका कि जो एक स्ट्रैंड होती है नई डीएनए की, वो पेरेंटल डीएनए की होती है. जैसे आप यहाँ देख सकते हो, ये येलो कलर पेरेंट डीएनए का ही है और सिर्फ एक स्ट्रैंड मैंने इस पे नई सिंथेसाइज की है. तो हमारी डीएनए रेप्लिकेशन जो होती है, इट इज अ सेमी कंजर्वेटिव प्रोसेस, विच मीन्स जब डॉटर डीएनए बनेगा उसमें एक स्ट्रैंड पुरानी होगी और एक ही स्ट्रैंड नई सिंथेसाइज की जाएगी. सो दिस इज द फर्स्ट प्रॉपर्टी ऑफ डीएनए रेप्लिकेशन, व्हाट वी कॉल एज सेमी कंजर्वेटिव नेचर, semi-conservative process. So first property we understood very well, that it is semi-conservative, which means that in the daughter DNA half the strand, one strand will be from the parental and only one will be synthesized afresh. Now property number second, it always proceeds in 5' to 3' direction. This is the second property, that DNA replication proceeds always in 5' to 3' direction. So you know that in a DNA double helix we have antiparallel helix. If this is a DNA double helix, it is an antiparallel helix. In this helix we have one strand which runs in 5' to 3' direction and another strand which runs in 3' to 5' direction. So during replication these strands will unwind. So these strands will unwind during replication. So unwinding results in separation of these two strands. So this is the 5' 3' strand and this is 3' 5' strand. Now when new DNA will be synthesized, जब नया DNA बनने लगेगा वो 5' 3' डायरेक्शन में बनेगा on the 3' 5' strand. तो यहाँ ये 5' 3' strand है, तो इस पे जो नई strand बनेगी उसकी डायरेक्शन यहाँ से, उसको यहाँ से 5' होगा एंड यहाँ से 3' होगा नई स्ट्रैंड को. तो अब वो सिंथेसाइज होगी इस डायरेक्शन में, यानी 5' से 3' डायरेक्शन में. ठीक है. लाइकवाइज ये स्ट्रैंड 3' 5' है, तो नई स्ट्रैंड जो बनने वाली होगी, क्योंकि एंटीपैरेलल डीएनए हेलिक्स होता है, तो उस नई स्ट्रैंड को 5' यहाँ से होना चाहिए और 3' यहाँ से होना चाहिए एंड सिंथेसाइज वो इस डायरेक्शन से होगी 5' to 3'. ठीक है. सो व्हाट डू वी मीन बाय इट प्रोसीड्स इन 5' to 3' डायरेक्शन? इट मीन्स जब सिंथेसिस होगी तो जो नई स्ट्रैंड बनेगी वो 5' एंड से 3' एंड की तरफ जाएगी, 5' एंड से बनते हुए 3' एंड की तरफ जाएगी. ठीक है. सो दिस इज द सेकंड प्रॉपर्टी, दैट इट ऑलवेज प्रोसीड्स इन 5' to 3' डायरेक्शन on the 3' 5' end of the on the 3' end of the separated strand. तो separated strand के 3' end से start होगी और 5' 3' डायरेक्शन में ये proceed होगी. Second property. Third property of this DNA replication is that it involves whole DNA. It involves whole DNA. Whole DNA. It involves whole DNA. So this thing has to be remembered. हमारे पास और एक प्रोसेस होता है, we have another process what we call as transcription. Transcription जो है वो एक स्पेसिफिक पोर्शन ऑफ द डीएनए का होता है, एक स्पेसिफिक जीनस का होता है, यानी एक उसमें एक पोर्शन ऑफ डीएनए इन्वॉल्व होता है. लेकिन इन दिस रेप्लिकेशन जितना भी लेंथ आपके डीएनए का है ये पूरा का पूरा इन्वॉल्व होगा एंड टू एंड. ठीक है. तो ये थर्ड प्रॉपर्टी हो जाती है इसकी, इट इन्वॉल्व्स द होल DNA. So we have three properties of this DNA replication. First property, this semi-conservative process which has been proved by Messelson and Stahl experiment, and it always proceeds in 5' to 3' direction. And property number third, it involves the whole DNA. Now we will discuss what are the requirements for this DNA replication. What are the requirements of DNA replication? What are the requirements of DNA replication? What do you need to replicate your DNA? What are the requirements of DNA replication? क्या चाहिए आपको DNA replication के लिए? सबसे पहले आपको substrates चाहिए जिनसे आप नया DNA बनाओगे, और वो substrates क्या है? Nucleotides है. And you should remember those nucleotides are used in their triphase phosphate form. ड्यूरिंग द सिंथेसिस। नॉर्मली होते तो वो मोनोफॉस्फेट फॉर्म में है, लेकिन इन डीएनए रेप्लिकेशन दे आर यूज्ड in their triphosphate forms. So first requirement is triphosphates of nucleotides. Triphosphates of nucleotides. तो कौन कौन सा ट्राइफॉस्फेट चाहिए आपको? आपको ट्राइफॉस्फेट चाहिए डिऑक्सी एडिनोसिन ट्राइफॉस्फेट, DATP जिसको आप बोलते हो। आपको डिऑक्सिटी गानिसिन ट्राइफॉस्फेट चाहिए, DGTP जिसको बोलते हो। आपको डिऑक्सीसाइटिडीन ट्राइफॉस्फेट चाहिए, DCTP जिसको बोलते हो। एंड आपको डिऑक्सीथाइमिडीन ट्राइफॉस्फेट, DTTP जिसको आप बोलते हो। ठीक है, तो ये आप फॉस्फोराइलेज एंजाइम से सिंथेसाइज किए जाते हैं। नॉर्मली इनके मोनोफॉस्फेट फॉर्म्स हमारी न्यूक्लियोप्लाज्म में प्रेजेंट होते हैं, तो उन्हीं मोनोफॉस्फेट फॉर्म्स को फॉस्फोराइलेशन करके 2 एक्स्ट्रा फॉस्फेट्स ऐड करके आपको ये ट्राइफॉस्फेट्स ऑब्टेन किए जाते हैं। तो याद रखोगे यहाँ पे दीज ट्राइफॉस्फेट्स आर ऑब्टेन्ड बाय phosphorylation of their monophosphates. Phosphorylation of monophosphates. तो ये मोनोफॉस्फेट्स की फॉस्फोराइलेशन से ये ट्राइफॉस्फेट्स बनाए जाते हैं। तो फर्स्ट थिंग इज द सबस्ट्रेट, आप जिससे नया डीएनए बनाओगे, एंड वो सबस्ट्रेट क्या है? दैट सबस्ट्रेट इज ट्राइफॉस्फेट्स ऑफ द न्यूक्लियोटाइड्स। ट्राइफॉस्फेट्स यानी DATP, DGTP, DCTP एंड DTTP। ठीक है, फर्स्ट रिक्वायरमेंट। नाउ रिक्वायरमेंट नंबर 2 रिक्वायरमेंट नंबर 2, दूसरी चीज आपको क्या चाहिए? व्हाट डू यू नीड? सेकंड थिंग। ठीक है, तो दूसरी चीज आपको चाहिए वो एंजाइम जो आपको इन सबस्ट्रेट्स को जोड़ के दिखाई देगा, जो इनको जोड़ेगा आपस में। एंड व्हाट इज दैट एंजाइम? दैट एंजाइम इज डीएनए पॉलीमरेज। तो सेकंड रिक्वायरमेंट इज डीएनए पॉलीमरेज। ठीक है, हमारे पास यूकेरियोट्स में और प्रोकैरियोट्स में डिफरेंट डीएनए पॉलीमरेज होते हैं। अगर हम प्रोकैरियोट्स की बात करेंगे, प्रोकैरियोट्स में हमारे पास 3 डीएनए पॉलीमरेज होते हैं। वी हैव डीएनए पॉलीमरेज 1, वी हैव डीएनए पॉलीमरेज 2 एंड वी हैव डीएनए पॉलीमरेज 3। 3 इनमें से मेन होता है, ये आप याद रख लो। एंड लाइकवाइज इन यूकेरियोट्स वी हैव सेवरल टाइप्स। यूकेरियोट्स में बहुत सारे डिस्कवर किए गए हैं, लाइक वी नेम देम अल्फा, बीटा, गामा, डेल्टा एंड एप्सिलॉन। ठीक है, ऐसे टाइप्स ऑफ डीएनए पॉलीमरेज हमें यूकेरियोट्स में मिलते हैं। ठीक है, तो डीएनए पॉलीमरेज डिफरेंट टाइप्स के हैं। तो इन शॉर्ट आप याद रखोगे डीएनए पॉलीमरेज एक ऐसा एंजाइम है जो आपको ट्राइफॉस्फेट्स को जोड़ेगा। कैसे जोड़ेगा? बाय फॉर्मिंग द फॉस्फोडाइएस्टर बॉन्ड, उनको फॉस्फोडाइएस्टर बॉन्ड से जोड़ेगा। ये जो ट्राइफॉस्फेट्स की एक्स्ट्रा एनर्जी है ये उसको यूज करेगा टू फॉर्म द बॉन्ड बिटवीन द एडजेसेंट न्यूक्लियोटाइड्स। तो दूसरी रिक्वायरमेंट आपकी क्या थी? डीएनए पॉलीमरेज। नंबर थर्ड रिक्वायरमेंट आपको क्या है? आपको सबस्ट्रेट भी मिल गया, आपको पॉलीमरेज एंजाइम भी मिल गया। ठीक है, इसके अलावा आपको प्राइमर्स जरूरत पड़ते हैं, जो प्राइमर्स सिंथेसाइज होते हैं बाय व्हाट वी कॉल एज आरएनए प्राइमेज एंजाइम। तो आरएनए प्राइमर्स आपको जरूरत पड़ते हैं जो एज ए स्कैफोल्ड एक्ट करते हैं, जो 3' एंड प्रोवाइड करते हैं डीएनए पॉलीमरेज को। आपका डीएनए पॉलीमरेज तभी एक्ट करता है जब उसको 3' ग्रुप अवेलेबल होगा, एंड वो 3' ग्रुप अवेलेबल कराना इन द फर्स्ट प्लेस आरएनए प्राइमर्स का काम होता है। वो डीएनए पॉलीमरेज को इन द फर्स्ट प्लेस सपोर्ट करते हैं, एंड दैट्स व्हाई आरएनए प्राइमर्स भी हमें जरूरत पड़ते हैं। तो थर्ड रिक्वायरमेंट इज द आरएनए प्राइमर्स विच आर सिंथेसाइज्ड बाय एंजाइम आरएनए प्राइमेज, जिनको आरएनए प्राइमेज एंजाइम सिंथेसाइज करता है। एंड लास्ट बट नॉट लीस्ट आपको अनवाइंडिंग एंजाइम्स चाहिए। ठीक है, एंड इनमें अनवाइंडिंग एंजाइम्स में आपको सबसे इंपॉर्टेंट है जिसको आप बोलते हो डीएनए हेलिकेज। इसमें आपको कुछ प्रोटीन्स चाहिए। यू ऑल्सो नीड, वी कैन ऑल्सो से अनवाइंडिंग सिस्टम। एंजाइम्स से बेटर हमारा टर्म यहां पे रहेगा अनवाइंडिंग सिस्टम, जिसमें आपका एक एंजाइम आएगा, हेलिकेज, प्रोटीन्स आएंगे जिनको आप बोलते हो SSBPs, सिंगल स्ट्रैंड बाइंडिंग प्रोटीन्स, एंड इसमें आपको कुछ एंजाइम्स आएंगे और जिनको आप बोलते हो टोपोआइसोमेरेज। वी विल सी द फंक्शन ऑफ दीज कंपोनेंट्स इन द प्रोसेस। तो फिलहाल हमने यहां पे बोला कि हमें 3 चीजें रेप्लिकेशन के लिए चाहिए: हेलिकेज, proteins called single strand binding proteins, and topoisomerase. So let us revise what are the requirements. The first requirement was triphosphates of nucleotides. Second requirement was DNA polymerase, which we have prokaryotes and eukaryotes में जो अलग अलग होते हैं, which are different in prokaryotes and eukaryotes. Here prokaryotes, third is the main one, but in eukaryotes there are various types. And we have RNA primers and we have unwinding enzymes which include helicase, proteins called SSBPs and topoisomerase. Now we'll discuss the process. Now we'll discuss the process of DNA replication. The process. So we'll discuss it simply in the simple means. So if you have this DNA, if we take this a portion of DNA, so what will happen first? This DNA portion will unwind at a portion. एक specific site होती है at a specific site with the site which is called as ori. एक specific site होती है जिसको आप ori बोलते हो, origin of replication. तो वहां पे क्या होता है, ये DNA unwind होता है. This DNA unwinds at this portion. And ये unwinding यहाँ पे इस enzyme से होती है, जिस enzyme को आप बोलते हो unwindase या helicase. DNA unwindase और helicase. तो ये इसको DNA को ऐसे unwind करेगा यहाँ पे. It will unwind the DNA like this. ऐसे. तो helicase एक enzyme है जो मैं यहाँ पर रेड कलर से दिखाऊंगा. तो यहां से भी हेलिकेज ने इसको अनवाइंड किया है, यहां से भी हेलिकेज ने इसको अनवाइंड किया है. तो फर्स्ट स्टेप इज अनवाइंडिंग. First step is unwinding. So what happens in unwinding? This DNA helicase operates on this double strand and breaks this double strand, breaks the hydrogen bonds between the nucleotides and separates this DNA at this site, what you call as ori, origin of replication. And ये जो structure यहाँ पे unwinded structure बनता है, this structure is called as replication bubble. It is called a replication bubble. This unwinded portion formed here is called replication bubble. यहाँ पे आपको ध्यान रखना है कि जो separated strands है, they can also again rejoin by means of these hydrogen bonds. So they are prevented from rejoining by these proteins, by these yellow colored proteins, what you call as single strand binding proteins. So SSBPs, single strand binding proteins. Single strand binding proteins. What do? They prevent rewinding of DNA. ये rewinding of DNA prevent करती है. एंड और एक चीज आप यहाँ पे याद रखोगे, जब ये DNA अनवाइंड बीच से ऐसे होता है हेलिक्स, तो यहाँ पे टेंशन आ जाता है एक्स्ट्रा, इन एंड्स पे एक्स्ट्रा टेंशन आ जाता है. तो एक्स्ट्रा टेंशन रिमूव करने में आपको हेल्प करते हैं एंजाइम इज कॉल्ड टोपोआइसोमेरेज. सो यहाँ पे टोपोआइसोमेरेज एक्ट करेंगे. एंड व्हाट विल दोस टोपोआइसोमेरेजेस डू? दे विल रिड्यूस द टेंशन at ends. And how do they do it? They do it by creating nicks, by creating cuts, temporary cuts in the DNA strands. Now how that is done, that is little complex, we will not go into that. Okay? We'll just remember that topoisomerases, they help in reducing the tension at the ends. Now look at this replication bubble. We'll observe that this replication bubble, it has two identical halves. It is symmetrical. The identical halves of these replication bubble are called as replication forks. You can understand it like this. So this is one half of this replication bubble, and this is another half of this replication bubble. And these identical halves of this replication bubble are called as replication forks. This is a replication fork, and this is another replication fork. Now replication forks, the replication on either side of the replication forks is same. जैसे डीएनए रेप्लिकेशन इस रेप्लिकेशन फोर्क में प्रोसीड करेगा, वैसे ही डीएनए रेप्लिकेशन इस रेप्लिकेशन फोर्क में भी प्रोसीड करेगा. वी विल कंटिन्यू द रेप्लिकेशन इन वन ऑफ द रेप्लिकेशन फोर्क्स. हम एक रेप्लिकेशन फोर्क में देखेंगे. सो इफ आई टेक दिस एज अ रेप्लिकेशन फोर्क, if I take this one replication fork, so consider this as 5' end of this strand, and this will be 3', and correspondingly this will be 5' and this will be 3' end. Now what will happen next? The RNA primase enzyme will come and it will synthesize RNA primers for the DNA polymerase to act. On this 5', 3' strand, the strand which has 5' and on this side its opposite strand will be having 5'3' direction on this side. So this will be 5'3' direction on this strand. Therefore, this primer will be synthesized right here. So this is the 3' end, and at this 3' end, the primer will be synthesized like this. And on this strand, ये जो opposite वाली strand है, इसका यहाँ से 3' end है, तो 5' direction opposite जो इस पे synthesized strand होगी वो ऐसे ही होगी, यहाँ से ही होगी, and इसका primer यहाँ पे synthesize होगा. The primer will be synthesized like this. So this will be the 5' end of the primer. This will be the 5' end of the primer. Now after that, DNA polymerase will start acting, and it will start adding the nucleotide triphosphates, and it will start synthesizing the strand. And likewise on this primer, this is RNA primer, this RNA primer. Likewise on this RNA primer, again DNA polymerase will act, and it will start synthesizing the DNA polymer on this strand. Now the replication, as the replication proceeds, this DNA starts unwinding in this direction. DNA is unwinding in this direction. So I will show it on the next slide. So DNA has unwinded further. Our previous primer was right here, and it was 5' end, and this strand was 5' on this end, and this strand was 3' on this end. So this was 3', this was 5', and our primer was right here, and our replication completed to this point. Likewise in this strand, our primer was right here with its 5' and right here, and this replication proceeded to this point. To this point. Now as you can see, this portion of DNA has unwinded further. This portion of DNA has unwinded further. Now for this strand, there is no 3' end available, or we can say there is no support here again. For there is no RNA primer, there is no 3' end available. So DNA polymerase cannot add the nucleotides now, because on this strand we don't have another 3' end available. DNA polymerase is capable of adding nucleotides to the available 3' end. Likewise जैसे हमारे पास यहाँ पे 3' end available है, इसका ये 5' है, 3' end है, तो यहाँ से DNA polymerase add कर सकता है nucleotides. So on this strand, as 3' end is available, तो DNA polymerase will continue adding the nucleotides like this. But on this strand we don't have 3' end available. So what has to be done? Another primer has to be synthesized right here, and the synthesis of this primer will take time. And after that primer is synthesized, then DNA polymerase will continue to add the nucleotides like this. So as you can see in this strand, DNA polymerization continued smoothly or continuously without any breaks. This is called as continuous strand or leading strand. This is called as continuous or leading strand. And in this continuous or leading strand, DNA polymerase proceeded smoothly or continuously. But on this strand, DNA polymerization is occurring in the form of fragments. You have to synthesize the new primers again and again. This is called lagging strand or discontinuous strand. And these fragments of DNA polymerization, in which you have to synthesize another primer again and again, these are called as Okazaki fragments. Then another DNA polymerase will come, which will remove these fragments and then resynthesize DNA on it. So then DNA polymerase 2 in case of prokaryotes, that will come and that will remove these primers. So there will be removal of the primers, and after that there will be resynthesis of the DNA. So there is removal of primer and resynthesis of DNA. There will be removal of and resynthesis of the DNA by another DNA polymerase, which is DNA polymerase 2 in case of prokaryotes. DNA polymerase 2 in case of eukaryotes. Okay. And this process will continue until it reaches the ends of the DNA, and then the replication will be completed. And when the replication will be completed, you will have the two strands like this, and new DNA like this. And hence you have these two copies of DNA. Two copies of DNA. Two copies of DNA. So this was brief introduction to the DNA replication process. I'll revise what I said so you can look back. So what I said, in the first place, there will be unwinding of the DNA at a specific point, what we call as ORI. This unwinding will occur with the help of enzyme, what you call as helicase. After the unwinding, there will be formation of this replication bubble. In this replication bubble, the strands will be prevented from resealing by these single strand binding proteins, and also the tension will be relieved at the ends by these enzymes called topoisomerase. This replication bubble, on this replication bubble, the replication will proceed bilaterally in both the directions. So DNA replication is bidirectional, and we'll study the replication in one fork. In one fork, we said, as DNA replication always proceeds in 5'3' direction, DNA polymerase always adds the nucleotides in 5'3' directions, so primers are to be placed accordingly. On this strand, the 5'3' direction will be like this, so primer will be placed like at this point of the unwinded portion. And on this strand, the 5'3' direction is like this, so primer will be placed at this position. And now DNA polymerase will add the nucleotides continuously in this pink color, you can see, and in this strand DNA polymerase will add nucleotides in this direction, like you can see. After that, the unwinding, further unwinding will occur, and this was a new portion unwinded. This was a new unwinded portion, as I indicated it, from this point to this point. And you could see that in this strand there was no 3' end available, so we had to place another primer. This was another primer, and this another primer provided the 3' end for DNA polymerase to act upon, and then this replication proceeded in the form of fragments. These fragments, I said, these fragments are called Okazaki fragments, and this strand in which DNA replication occurs in the form of fragments is called as lagging or discontinuous strand. The opposite strand, on the opposite strand, there is always availability of the 3' end, in which the DNA replication will proceed in 5' to 3' direction. So that strand we don't have to resynthesize primers again and again. So that strand will become continuous or leading strand. That strand will become continuous or leading strand. And this thing will continue. And meanwhile, I said the primers will be removed by another enzymes, another polymerase, like we have this DNA polymerase 2, which removes the primers and adds the nucleotides, removes the RNA primers and adds the nucleotides of DNA in place of them. And the ends will be joined, where we have these nicks. There will be nicks at the points, and those nicks will be joined by DNA ligase. Those nicks will be joined by DNA ligase. And this process will continue until the whole of the DNA is replicated. At last, you will be having 2 copies of the DNA. So our IT head, Mr. AIJAZ Ahmad, will close this topic. That will be end of the DNA replication.